Okay, dokie. So let's get into chapter 11. We'll start with part A. And chapter 11 is just really talking about the foundational stuff of the nervous system. And so we'll talk through that. Part B also talks about the basically generation of electrical signal and how we do that in the nervous system vis-a-vis a neuron, how that works as well. So we'll discuss that. And then chapter 12 then starts talking about the brain. And so we'll get into that hopefully on Wednesday, Thursday, okay? All right, so again, we start off with the general kind of orientation of the nervous system and how that really functions. And so we look at an overview and then we get further into some of the details, right? So as we've already established this semester, right, the nervous system is the controlling center, and it's actually the one that's overseeing all of the other systems of the body, the whole entire system of the body. The whole organism, in fact, is through the nervous system. The nervous system is the one that's really regulating everything throughout that whole 11 body systems, right? And so we do this through communication, right? Through a stimulus and through a receiving of that stimulus, and then through that processing by the nervous system, and then through that effect or response to what they're stimulating, right? So we do this in two ways, right? We've seen that the nervous system has electricity, right? It can generate electrical energy, right? And we also have seen, especially in the muscle tissue, that the nervous system also sends a signal out through a chemical, right, or chemicals, right? And so when we take a look at, for example, if I was to draw then the motor end plate, okay, and down here I would draw the muscle, right? And we saw, for example, that we saw an action potential arrive at the motor end plate or the synaptic end bulb or whatever the end of the neuron would be, right? This would be considered the electrical signal, right? And if you recall the synaptic vesicles, those would be my synaptic vesicles, each containing several molecules of a chemical we call acetylcholine, right? And we saw that acetylcholine migrate across what we call the synaptic cleft and then get received by a receptor. We know that this side then, this right here, is the chemical signal. Does that make sense to you? Both released by the neuron, both carry a signal. It's just a different signal from here to there. Because that signal told those little, calcium channels to open, which then told the synapses to go to the end and release their product, which are the chemical signals for the muscle. So both of which are used by the neuron for communication and control, right? Rapid and specific. It's an almost immediate response when we talk about rapid, very, very fast, right? Very fast signaling of the nervous system. Okay, so we've talked about these before when we talked about, for example, homeostasis at the beginning of the course. And so we still use these, and in fact we use these directly in the nervous system because the nervous system really has two main parts, right? Or three main sections, if you will, or responsibilities, right? The nervous system can act as a sensory side, right? So there are parts of the nervous system that are responsible for sensation, right? Sensing things, right? From the body. We can commonly think of that as gathering information from all parts of the body or the environment and the environment, because we're taking this information and we're going to send it to a part of the body that's, again, going to process the information and make decisions about that information it has received, right? And so when we talk about internal, external changes, that's exactly what we're talking about. We're talking about taking this information from sometimes outside the body, at other times inside the body, or sometimes at the same time, and just gathering that information and sending it to the next part where we talk about the integration part, because the integration part is the processing place. It's going to process the information. It's going to interpret the information, and many times it's going to take that information and respond to it. After it's processed, interpret it, then respond in some way to that information, to that sensation, if you will. And that's really the third side, is the motor output. And the motor output is talking about the effect. What is the effect of the sensory integration, and then what is the output, right? The output is always going to be a muscle or a gland of the body. And notice it just says muscles, right? We don't specify, but this can very well, in fact, be cardiac, smooth, or even skeletal muscle, which we know to be found in all different parts of the body, and glands, right? Sometimes these are endocrine glands. Sometimes these are exocrine glands, which we know what those are, what the definition of an endocrine versus an exocrine gland. We've discussed that in the integumentary system. And we know for a fact, if I go back to the integumentary system, that there are several different types of exocrine glands. And I'm not going to list, but you know those, right? Oil gland, sweat gland, types of sweat glands, modified sweat glands. You know, that's what we're talking about, right? And the endocrine glands are part of the endocrine system. This is actually hormones, right? It'd be hormones. But that's really what it means, right? We're getting a response from whatever we have sensed or gathered information from. The brain has processed it and interpreted it and then made a response. And many times that response is going to change the stimulus. It's going to change the information that we've gathered, in fact. Okay? So you got to keep these three in mind, because when we discuss some of the details, it's always going to be, where does this fall in fact? Or how does this play a role in one of these three or all of these three? Okay? So there's a quick example, figure 11.1, that you find in your book. Talks about the sensory side versus the integration side versus the motor side. Obviously, sometimes our sensory receptor can be really, really specific and really quite unique, like that. And even at that, we call that special one a special sense, right? Eye would be part of the special senses. That would be part of the nervous system. Really, really important one, because it gives us vision, right? We can see things. So everything that comes into our eye, including visible light, is going through our eye. Attached to it is a receptor sending signals to the brain, right? That brain is going to then process it, interpret it, maybe recall it from a previous experience vis-a-vis a memory, and then it's going to do something about it, right? And here's the innervation, or the motor output, or the effector. And our effector here is our biceps brachii muscle. And that biceps brachii muscle is going to contract, thereby moving our hand and drinking water, let's say. Okay. There's our three functions. Okay. So let's get more specific into the nervous system, talk about that. The nervous system is divided into two main parts. The central nervous system, or the CNS, which includes the brain and the spinal cord. This falls on number 2 of those functions, the integration part of the functions. And the other section is the PNS, or the peripheral nervous system. And this is pretty much anything outside the CNS that's not brain or spinal cord would be considered PNS. And so there's two main sections of the PNS, the spinal nerves and the cranial nerves. Easy to remember because the spinal nerves stand for spinal cord, so these are the nerves coming out of the spinal cord. So spinal cord is CNS, but anything kind of pushing itself out is considered spinal nerves in the spinal cord. And the cranial nerves are anything that comes out of or going to the brain. Okay? As I mentioned, the eye is connected to part of that relay system. That would be the optic nerve. And in fact, the optic nerve is one of the cranial nerves that we have that connects the eye to the brain, as an example. And actually, in fact, there are 12 cranial nerves. And we'll go through that next week. We'll talk about those. Okay? So this figure, figure 11.2, which is also found in your book, shows you what is peripheral or central versus peripheral, what falls in that list there. Okay? That right? I would put this as the somatic and the autonomic, right? And I would say that this would be skeletal muscle and then voluntary, right? So you build the chart. And in fact, there's a figure for that you're going to see here in your book and also here in the PowerPoint. Both somatic and autonomic are both gonna feed to muscle and glands, right? It's just now we're defining which type of muscle depends on which type of nervous system it's feeding. Because again, this is also PNS. And in fact, we can also put, this would be motor too. And I forgot to put that down on the somatic side, but that's exactly where we're at. Remember I told you that there was two main sides and then the integration center in a nervous system, right? Think of the sensory as input and think of the motor as output. Because that's what the signal or the information is coming from. Input inside or output, it's going out. The autonomic nervous system and the somatic nervous system are part of the output, right? Where are they outputting? The autonomic is outputting to visceral tissue, and in fact, it's the smooth muscle cardiac muscle, and glands. You see that it's not skeletal muscle because that is in fact, a voluntary control. It's a different type of muscle. Smooth and cardiac are involuntary, and the glands, of course, are involuntary as well. I mean, if you classify them, if they're voluntary, involuntary, we cannot control our glands, right? Those are automatically controlled. And that's why we call it the autonomic nervous system, automatic, right? Again, involuntary. And that one has some subdivisions there. We call that the sympathetic and parasympathetic. And we'll talk about those in more detail as we continue on, right? Here's the figure I mentioned, figure 11.3. And in fact, you're seeing all of the breakdown between somatic and autonomic. You're seeing the breakdown between what is sensory and what is motor in the peripheral nervous system. Okay? If you notice, the red arrow indicates output or motor, and the blue indicates input or sensory. Both of which fall under the peripheral nervous system, both of which are part of that, right? It's just where is it at? Now you'll notice that the blue doesn't go that far. There's not much to the blue. Pretty simple, in fact, sensory. All right? Gathering information from nerve fibers found on the somatic and the visceral, gathering information from the inside, the outside of the body, sending that signal to the brain and spinal cord. Brain and spinal cord, again, makes a response or processes the information, interprets the information, and sends a response. And the other side, the motor side, it depends on where it's sent and what it does, right? That's why there's a lot of red, because the motor side gets divided into somatic, the skeletal muscle, voluntary, or the autonomic, the automatic stuff, smooth muscle, cardiac muscle, and glands. And then, what is that response going to do? Depends on the division. If it's sending it for the sympathetic or the parasympathetic, we're going to see two opposites there, in fact. Okay? We know the sympathetic to be the fight or flight response, because that's what the response is going to be towards your body, right? And we classically use this in biology, right? Because I mentioned biology. I mean, we talk about human biology here, but biology in general, because this part of the nervous system is very, very old. What I mean by old is that it exists even before we were humans, right? When evolution, when we became humans, this has been around even before we became humans. This is actually in place with pretty much all animals, right? That's how old it is, in fact. When that signal comes here, it's going to direct the body either to do one or two things. This is commonly what we think of, but there's, it's not always fighting or flighting, but that's kind of what you see in animals. That response is going to be either going to fight or you're going to fly away or run away, right? Fight or flight response. This is the one that actually puts in energy, because energy is required to fight or flight, right? In fact, you see this in animals like lions and bears and tigers and big animals like that. You sometimes see that in fish, you see that in birds, you see that everywhere. They're going to defend their territory, defend their offspring, or in some cases fly away and survive, or run away and survive. Okay? And in fact, that still exists in our body, right? It exists in our body. We run into a confrontation, we see somebody that well, for whatever reason, we're either going to fight or flight, and many of us will prefer one over the other, right? Many of our families have the fight or flight response. Some of them will get angry in confrontations and fight or flight, but that's the response that's going on in our body, and that falls under the autonomic nervous system. That's actually what occurs there. This one is known as the rest and digest. Or sometimes, which is also my favorite, is the feed or breed response, right? Because that's what's occurring on the other side. This is the opposite of that. This one requires energy, so there's going to be a lot of energy getting used, made and used. This other one, not so much. We're turning down things. We're resting and digesting. And in fact, when I talk about feeding, right, eating, that's actually the signals that are getting sent to start that process. Breeding, right, reproducing in the same regard. Many of our processes are sent for the parasympathetic. Okay? All right, so that's the breakdown of the nervous system as far as how it's broken down, just generally speaking. And now we're going to talk about go down to the cellular level. So we talked about some overview, general kind of nervous system stuff. Now we're going to jump into cells, right? So we know what a neuron is, right? Right? A neuron is at this point we called it a nerve cell. And in fact, that's one of the main important cells that then carries that capability of generating and transmitting an electrical signal, right? But what we have not actually discussed is the other supporting cells we call neuroglia, okay? Or glial cells, in fact. These very much surround and wrap these neurons, and they're the supporting characters for the neurons and their work, in fact, because that's what they're going to do. Okay? There hasn't been historically enough attention placed on the neuroglia in science when we did research. We've always been researching neurons and how neurons work and how we can repair them and how we can grow them or how we can make more neurons. So there's been a lot of research on that primarily. Over the past, I want to say 20, 30 years or so, we started to look at the neuroglia, the little small cells that are the supporting cells. And lo and behold, when we started to take a look at those, we found that they are just as, or in many cases, more important than the neurons. Because the neurons, again, send the signal, electrical signal, action potential, if you remember that, right? And yes, they're vitally important. That's their main role. That's what the nervous system does through the neurons. But the neuroglia actually maintain the environment around the neurons, maintain the health of the neurons, repair the neurons sometimes, defend the neurons. They do all of those jobs, but they don't transmit electrical signals. It's what they don't do. So when we started to look at them, we found that they're crucially important. And when you damage those or take those away or they're abnormally functioning, bad things happen to the nervous system. So that's kind of where we're at as far as, brain research and nervous system research. So we've got 4 main neurons that actually support the CNS functioning, the neurons doing their job. Here's a list of them there, right? Astrocytes, microglial cells, ependymal cells, oligodendrocytes. Okay, we'll discuss each one of them. Here is our astrocyte. Most abundant, so one we're going to have in most numbers. Does a lot of different things for our body. Highly versatile. And highly branched, which means they branch out. Okay? So the reason we call them astrocytes is because astro is the root word, meaning star, right? Because they're very much of a star-shaped, large cell. So where do you find them? Clinging on, actually attached to neurons. The endings of neurons we call synapses, and then attached to capillaries or blood vessels. So talk about versatile. Here's the function or functions, in fact, of astrocytes, what they are responsible for. Okay? So notice that they support, they brace, okay, neurons. And when they say plain exchanges between capillaries and neurons, right? Blood vessel exchange. Because the blood vessels are capillaries, and so they're going to drop off nutrients and oxygen, and the astrocytes are going to kind of carry that through themselves to the neurons. And the neurons will create waste too, and that waste will be carried out through the astrocytes. They support, in fact, young neurons, right? So neurons that are just developing, astrocytes will kind of, take care of them for a while. The chemical environment around neurons are, astrocytes are responsible for that. We see those functions there. And here's a figure 11.4a is a picture of an astrocyte, very large, star shape. You can see it attaching to a capillary in many different places, and then you can see it attached to the neuron in the background, right? So in the foreground, you see the capillary, the astrocyte, and then attaching to a neuron. And there's the go-between. There's where the neurons and the capillary, the astrocytes, found. Okay? Next one is our microglial cell. By the way, I didn't mention this, but why do we call it glial cell? Because glio comes from the root word meaning glue, right? Because that's what they do. They stick, right? They stick onto neurons. They kind of hold stuff together. That's what we call them, glial cells, glue cells. The microglia are very small, oval shape. That's ovoid, which is an oval shape, not exactly perfect like that, right? You can see different shape sizes of the oval, but very small. It's got thorny processes, right? Little ends, the sides and ends come out to have these little look like thorny structures. Those touch neurons, they monitor neurons, their viability, their health, their overall functioning. They will be found surrounding injured neurons because that can occur as well. And then they have the capability of eating away or phagocytizing microorganisms, or breaking down and destroying or processing the debris from a neuron. Okay, debris is like pieces sometimes that won't get used, or if it's damaged, all the broken pieces get kind of processed by the microglia. So here's our microglial cell. You see it's much smaller than a astrocyte and much, much smaller than a neuron, because they actually put, you know, size relationship here as well. You can see how big because it's not all the neuron, but you can see how big the neuron is here. Just looking at its size. So if you took you could measure that, you would measure that, right? And look how small that is. Look at the size of that, right? In fact, I mean, you can measure the very ends of it, but still very small. All right, so you can see the thorny processes there. This is a thorny process, right? See those coming out. Little edges there, thorny processes. And they're physically touching the neuron, measuring neuron's viability, health, you know, so on and so forth. Okay, the third one is the ependymal cell. Ependymal cells can change shape. They can go from squamous, flat cells to columnar cells. So they can have different shapes. Some of them can be ciliated, which means at the very edge you have cilia or extensions that look like little kind of hairs on the edge of the ependymal cell. For example, the ependymal cells that line certain parts of our brain have those cilia, and they in fact help to move what we call cerebrospinal fluid. Because remember, that's a type of extracellular fluid. So here's your typical ependymal cells, right? And you can see these are in fact columnar here in this example. And you can see the cilia here lining part of the brain that has this fluid-filled cavity that then moves the fluid a certain direction, right? So they can act as barriers as well, right? And the last one here of the CNS are oligodendrocytes. These are also branched. They wrap around the nerve fibers in the CNS, and they form, through their wrapping, myelin sheaths, right? Myelin sheaths. Here you can see 3 oligodendrocytes wrapping around not only just one type of neuron, but several, right? So, you know, we'll just look at that one. You can see it wrapping here, there, there. So not only 3 different types, right? 3 different neurons, and in fact, a certain part of the neuron we call an axon, which we'll get into all the parts of a neuron, but you can see that one oligodendrocyte wrapping in three neurons. And in fact, the middle one is wrapped in 2 places. So it can be various different places. For example, you can see this one just wrapped one to each neuron, right? Just one time. Okay. Okay, so let's switch over to the neuroglia and the PNS, and there's only 2 there. So a total of 6, right? 4 in the CNS, 2 in the PNS. These 2 in the PNS are called satellite cells and Schwann cells. Okay? Satellite cells surround the cell body in the PNS. So here's your typical neuron, right? This area surrounding here would be considered the cell body. And it is in fact where you find the neuron. Excuse me, the nucleus, right? So the Schwann cell is going to surround the cell body. I'm sorry, the satellite cell is going to surround the cell body. And the Schwann cell is going to surround the area that we call the axon. Okay, Schwann cells behave almost exactly like astrocytes in the CNS. That's their complementary cell. They are not astrocytes. They function almost exactly like astrocytes, except we call them satellite cells, and they're in the PNS, not in the CNS. And the Schwann cells act like, as far as their function, oligodendrocytes. Even though they're not oligodendrocytes in the PNS, we call them Schwann cells. Okay, so figure 11.4e shows you then the purple cells, and those are satellite cells. You can see them surrounding just the cell body. And the blue cells surround the axon, the extension of the neuron. Notice they do not surround the cell body, and they do not surround the synapses. That's the very edge, or the end of the neuron. Only the axon. Okay, so those are the neuroglia. Now we've got the neuron, right? Okay. Neuron or nerve cell, structural unit of the nervous system. It conducts those electrical signals or the nerve impulse. That's another way to say it. It's got really special characteristics that are specific to them. Neurons or nerve cells can actually last a very long time, and in fact can stay alive for a person's lifetime. Most of them, the majority of them, are amitotic, which means no mitosis, right? That's what amitotic means, no mitosis. They don't undergo mitosis, which in fact is kind of something that we sometimes have to deal with, because if they're amitotic and they get damaged, then they don't undergo mitosis, right? They cannot divide, unfortunately. And in fact, most of your knowledge learning, right, what you've been learning so far this semester, let's say as an example, doesn't come from you making more neurons, because obviously that doesn't work that way. It's from making more connections of those neurons. And in fact, that's how you grow your knowledge. The connection of neurons between themselves. More neurons get connected, the more knowledge you have, because there will be more brain activity. There's more places to store things. This is why you see knowledge increasing. And it's not because you multiply brain cells. It's a common misnomer. It's a common misconception that people think that you're dividing, making more brain cells. And in fact, you're not. You're just connecting them more closely together. Okay? They have a very high metabolic rate because they have a continuous kind of function. They send signals continuously, okay? And they receive things continuously, and they're sending out chemical signals continuously. Because of that, they need to make all of those things. So there's a lot of energy involved in doing that. And that's why they need a continuous supply of oxygen and glucose, because they function primarily on glucose. That's what they prefer. And they also use oxygen, both of which are used to generate ATP, right? That's something that we've seen before, right? Does that make sense to you, right? Okay. Which is also really important to understand, because if you take away oxygen, or you take away or don't have enough glucose, then the first thing that's going to happen is that your brain is going to tell you it needs oxygen and glucose, or the neurons are telling the rest of the body that they need oxygen and glucose. That's what you see when you starve your body of oxygen, or starve yourself from glucose, right? Those neurons are telling you, hey, we need that stuff so we can continue to work. Okay? All of them have cell bodies, with the exception of one or two more processes, right? Little divisions, if you will, right? So the body is the perikaryon, or the soma. We call that the cell body of a neuron. Of course, you know that the center of a neuron will have a nucleus, and that nucleus has the DNA of the neuron, and will dictate, based on what it's read, many things, right? So a neuron is going to make proteins. They're going to make membranes, or membrane. It's going to make chemicals, all those things that a neuron needs to do its job every single day consistently is getting made in the cell body. There's a lot of rough ER there. So if you were to look at a neuron up close, you would see a lot of rough ER. And rough ER is actually endoplasmic reticulum with ribosomes attached to them, which means it's going to produce a lot of those proteins. A lot of lipids like membranes. A lot of other chemicals, and many of those chemicals are protein-based or amino acid-based chemicals. Okay? In a cell body, or in a neuron, the plasma membrane is going to, in the cell body, going to be the receptive region. It's going to receive input from other neurons. And in fact, this is what we're starting to see, how neurons are connecting to others. Because the edge, or the end of the neuron, from neuron, let's say number 1, is going to innervate or connect or touch, if you will, the cell body portion of the next neuron. And in fact, that's what we call it, the receptive region. That's where the signal's getting received from other neurons sending signals, or chemical signals, in fact, to the cell body. It processes that information and then will create and send a signal down to the edge of itself to a next neuron, and so on and so forth. So we have different terms depending on where the cell bodies are located. Okay? Depending on where the cell body is located. Most cell bodies are located in the CNS, and we call what we would call those clusters, or a bunch of the cell bodies kind of huddled around together, nuclei. Even though that sounds like a nucleus, but actually a nuclei if it's in the CNS. So they're going to be portions of the spinal cord and portions of the brain where you're going to find the nuclei, like basal nuclei, and so on and so forth, because those are the places. And so when you hear nuclei, when we talk about the brain, these are clusters of cell bodies kind of in one area called nuclei. If they're found outside the CNS, we call those ganglia, right? Ganglia. Many of them are found directly outside the spinal cord. And there's so many kind of put together that we've got a little kind of, looks like a little swelling or outpouching of the spinal nerve. We'd call that a ganglion. Okay. All right. So outside of the cell body, excuse me, we would call those processes, extensions outside the cell body. Okay. Extensions of the plasma membrane, in fact. In the CNS, the CNS contains both, right, the cell body itself and its processes or the extensions, right? And mostly in the PNS, we've got just the neuron processes. That the cell body is not part of that, right? So for example, if we talk about a spinal nerve, a nerve that comes all the way down, let's say your forearm, that is the process or the extension of the neuron itself, and it's not the cell body. Cell body for that would probably be located way close to the spinal cord, in fact, or sometimes in the spinal cord, depending on the spinal nerve. Okay, we'll see what that means, because I know that can be kind of confusing, but we'll kind of try to define that even more. Okay. So what is a tract? In fact, a tract is a term we use just for the CNS, and these are bundles of the processes in the CNS. Bundles of the processes. Okay? So again, that's what that means, right? Nuclei, cell body collection areas in the CNS. And if you're a tract, processes, right? In the CNS. Right? Okay, that's what that means, right? And I can write the same thing for the PNS, right? So in fact, nerves are bundles of processes for the PNS. That is when you talk about a nerve, right? A nerve of the body. Bundles of the processes of many neurons bundled together in the PNS. That's what we call the nerves, if you will. Okay? So what about these processes? What do we mean by processes? Why the word processes? Because they actually can be defined in two ways, right? Processes can be dendrites, or processes can be the axon of a neuron, or dendrites of a neuron. Okay, so let's look at figure 11.5 because it shows you everything that we just talked about, pretty much, except for the names for the CNS, PNS. But here's your typical neuron, right? All of these that we are extending out of the cell body are all dendrites. So we use the word dendrite to describe the extensions of the processes outside the cell body in a neuron, right? All of those. So I'll erase it all because it looks kind of confusing. But yeah, you see all those little extensions out of the cell body. That's not the cell body. So then I'll just draw the cell body. That's the cell body, right? The extensions out of those here, those are dendrites. One special extension out of the cell body here is called an axon. Okay? That is our axon. One special process that extends all the way out is called an axon. Okay? So those are the three main parts, in fact. The cell body, dendrites, axon. At the very end, we don't consider those really parts of axons, but we call those the axon terminals, little ends that intersect. And in fact, you would probably see another neuron here. Right? It's connected to the neuron, or going to then signal to the next neuron. So they're all basically interconnected. The more interconnections you have of all the neurons, the more they can process information really quick, or recall information very quickly, or store information really quickly and efficiently. And in fact, that's what knowledge is all about. That's what having a brain is really all about. That is how we actually learn new tasks, is interconnections. More connections that are made, in fact, the more knowledge you have, the more you can process things, so on and so forth. Okay? So, a little bit about the dendrites, right? In the motor neurons, we've got hundreds of these little dendrites. That's just to give you an example that they can be many, many. But this is the input region of the neuron, right? Signals come from dendrites to the cell body. And in fact, the electrical signal, the nerve impulse that we have here, coming from the dendrites to the cell body, and we call those specialized little signals, graded potentials. Graded potentials. We've heard of another potential. We call that other potential an action potential. You remember that one from muscle tissue, right? That's the one that comes down to the end of the neuron. And then chemical signals received, and then that opens up channels in the muscle, and then the muscle creates its own action potential, right? But that's not what we're talking about here. But I just wanted to mention that we've already talked about a type of potential. And this one's an action potential, but this one here, the short distance between in a dendrite sends a signal to the cell body is called a graded potential, not an action potential. When you look at neurons that are found in the brain, many of those areas have these neurons that have dendrites that are really fine, right? They become thinner. They can actually become even more processed or pulled out, right? Specialized neurons that are found in the brain that have these are called dendritic spikes. That's the word to describe it. Because they actually look like spiky ends. They kind of look like microglia if you look at that, right? It's figure 11.5b in your book, right? But you see that there. Can you see how these long strands, in fact, are dendrites? Right, those are all dendrites. But then the little bulbs at the very end that kind of look fuzzy, right, on the dendrite, those are called dendritic spikes or spines. And this would be for pretty much in the CNS. This would probably be brain neurons. Highly sensitive. By making them really small and making those little bulbs, right, or spikes or spines, then they'll collect really, really small amounts of information, really precise information. Okay. So let's talk about then the axon, right? The extension of the cell body, right, of one axon starts at this cone-shaped area, and we call that area where it's starting to form the axon hillock. Not all brain cells have axons. Some are missing. They don't have an axon, right? Some of them do, and they're really short. And some of them are really, really long. So there's a combo depending on what what neuron is doing what. So not all of them have an axon, just to kind of just tell you that, right? Look at that. There's an extreme case, right? There's certain ones that are at that length, right? The axon is 1 meter long. That's a little over 3 feet, by the way. It's a really long axon. And where would you probably see that? Those axons that they're talking about, those neurons that they're discussing, are sometimes motor or sensory neurons that extend, for example, at the small of your back or the lumbar sacral area. And then those neurons will come from there and go on, travel all the way down your thigh, your hip, all the way down to your leg, and then end up, innervating places down your leg. And if you measure that, that's, can be about 1 meter or so. That's how far some of those axons will travel. Long axons are called nerve fibers. Sometimes they branch, and these are called axon collaterals. Again, these are definitions. The axons at their very end will branch, and we call these terminus or termini, right? Axon terminals or terminal boutons are sometimes what they're called, right? Some can be decayed. The axon can be decayed or damaged from that decay or that cut of the axon portion. Right? Okay, so again, I mentioned there's a lot of movement, right? It's not like the stuff just falls down to the very edge and then, falls down to the other side of the edge. There's a lot of movement occurring. And in fact, back to this organelle, right? Cytoskeleton, in fact. Cytoskeleton gives shape to a cell, but it also participates in movement. So there is movement occurring from the axon to the cell body and the dendrites, or from the dendrites to the cell body to the axon. And we talk about these two words being used to describe the away from the cell body or towards the cell body. Anterograde versus retrograde movement. Okay. Again, the axon and the terminal end need things to be, proteins that need to be renewed. If we've released a bunch of, let's say, neurotransmitters, you need to replace them, and they're going to come from the cell body and get transferred down to the end. Or you need to take stuff back, you can. So that will occur. And in fact, when we look at this, just a little microbiology, right? Some of the particular bacteria, sometimes bacteria secrete toxins, or sometimes viruses, can use the cytoskeleton as a transport mechanism to transport their way up and down a neuron. So things like polio, for example, which is a virus, rabies, which is a virus, herpes, which is a virus, and tetanus, botulinum bacteria can secrete a toxin, and that toxin can carry its way back and forth, right? So the virus can travel up, or the virus can travel down, and that's why we see, for example, transmission occurring, right? Infection and transmission from one neuron to the next. And in fact, that's how polio happens. Polio can actually, many different ways, but the main way is to breathe it in, and then the neurons that are connected to your lungs can catch it, and then the virus will go up and then damage neurons. Right? Okay. So let's talk about myelin sheath really quick, right? Myelin sheath, the reason we call it myelin sheath is because it's a chemical composition called myelin, and that myelin's a protein lipid substance. It's a white substance, and in fact it surrounds the axon. Many axons get surrounded, protects the axon, but it also insulates the axon. For example, if you have electrical cords are wrapped with plastic, in fact, special type of plastic, and that actually insulates the electricity, because as the electricity goes to the wire, it concentrates it, focuses it to the area it's supposed to go, and if you didn't have insulation, the electrical signal would shoot out everywhere. And in fact, that's what, if you think of it that way, the myelin sheath works in that regard, right? So some axons will have myelin that would be called myelinated fibers. Some axons do not, and we call those non-myelinated fibers. Again, it just depends on where they get used at. If it's a PNS neuron, then the Schwann cells, not the satellite cells, but the Schwann cells in the PNS are the ones that form that myelination. So here's your typical PNS. I don't want you to study the steps here, but just to show you that this is one Schwann cell. That's one Schwann cell. And you can see it how it's kind of enveloped around the axon. And we'll start to roll and roll and roll. We call this a jelly roll, right? But that's kind of, you know what a jelly roll is. That's what a jelly roll is. It'll wrap itself around and around and around. And it forms then a myelin sheath, right? And in fact, you can see this here. This is a electron microscope. 24,000 times, right? And you can see this really dark area, and if you see really close, you can see the lines, right, of its plasma membranes, you know, surrounding around and around and around. That's one myelin sheath. It's a cross-section view, and you can see the internal part of the axon. In the PNS, you're going to have these little gaps in between the myelin sheath. So it's not completely myelinated. There's going to be spaces in between the axon that don't have myelin. We used to call those nodes of Ranvier. I don't think that gets used a lot anymore. In science books, we call them myelin sheath gaps. Right? You're going to see how those work also shortly in the next part of the PowerPoint. Okay, so again, just to show you this figure here, we see that there, myelin sheath gaps. In the CNS, those are formed by the oligodendrocytes. And in fact, it's not, again, oligodendrocytes can be one cell surrounding many of them. And so we've seen this before in research, right? We've seen that some oligodendrocytes can wrap 60 so far. I mean, we don't know yet. It could be more or less, or it could be more than 60 axons, but that's what we found out so far. That one neuroglia can wrap up to 60, or a couple. So just on some anatomy here, right? In the CNS, the places in the brain and spinal cord, because that's the CNS, right? The brain and spinal cord. We have collections of myelinated fibers. You would call those white matter, right? White matter. And the places that you don't have myelin, which obviously the cell body doesn't have that, or some neurons that don't have myelin on their axon, we call that area gray matter, white matter, gray matter. And you'll get to see this again. We'll, in fact, dissect a spinal cord and a brain, so we'll see both of those, right? Gray matter and white matter, you want to point those out, right? So again, here's your typical oligodendrocyte. Again, there's your same, the same figure, but now we see that the oligodendrocytes make the myelin sheath, and then there's also gaps in between as well. Okay. Finally, before we get to part B, this is the last part of this part A, we actually are going to group neurons depending on their structure, right? What do you mean by structure? The processes, right? What extensions do they have outside of the cell body? Okay. If you're multipolar, you've got 3 or more processes. One of them is always going to be the axon. The other 2 or more are going to be the dendrites. That would be considered a multipolar neuron. A multipolar neuron is the most common and the major type in the CNS. Most of your brain and spinal cord are going to be multipolar. Again, you have more connections, more signaling, more interaction with other neurons, and this is what's going to happen in the brain. Bipolar are just going to have 2 processes. One of them is always going to be the axon, but they're only going to have 1 dendrite. We don't have them a lot in our body, so this one is not very common. So they give you 2 main areas, right? 2 main places where you'd find them. The retina, for example, and the olfactory mucosa, which is the area where you're going to have the start of the neuron that's a bipolar neuron. Does anybody know what olfactory means, by the way? What does olfaction mean? Sense of smell. That's olfaction, right? These are going to be located at the edge on the membrane, mucosal membrane of your nose. And in your mouth as well, because you can also taste or smell from your mouth, believe it or not. And in fact, those are bipolar neurons that are all connected there that are going to feed information to the brain. Same thing with your retina, that's the eye, of course. And finally, the unipolar neuron. It's always going to have two axons. Always going to look like a T shape, although the T shape can be, a straight T, sideways T, sometimes upside down T, right? As far as when you're looking at them, but T nonetheless, right? So here's a breakdown of each one. There's table 11.2-1, and you see there's your typical multipolar neuron. That's the one we saw before. We just didn't know it was a multipolar. And then there's the other one, the bipolar. Two sides to that, and then the unipolar neuron. So they can have different types of formations depending on where they're found. So sometimes you can see this, right? These are both multipolar, but in fact they just have one axon, although there's a branch there, but that doesn't classify as an axon. And here they only have one, looks like 2, 3 dendrites. Even though everything else outside of that here is different than, let's say, here. Because everything I drew there are considered dendrites. If it's either 3 or more processes, one including the axon, is considered a multipolar, right? And look at this one. This is a bipolar. These are 2 bipolars, in fact. 1 axon. This is considered 1 dendrite. Even though you see this kind of weird extension out there, those don't count. Or you see this weird extension over here, this is the only one that counts. 1 and 2. Bipolar. So why is this one different than that one? It's because you see in fact, 3 dendrites there, and this is only 1 dendrite there. Whatever comes out of the cell body is what you consider counting towards that number. And here's the unipolar, is the T shape, but the sideways kind of T there, where you have 2 axons. One going this way, one going that way. Unipolar. Okay? Now, again, there's the functional classification, right? What's considered sensory, motor, and integration side. But now we're looking at neurons, right? Types of neurons. Unipolars are almost always sensory. Unipolars are that upside down T or sideways T, the cell body right in the middle, or the cell body kind of up here, forming that. Almost all of them are unipolar that are sensory, or sensory are all unipolar. And the cell bodies are located in the ganglia of the PNS. Okay? Now sometimes they can have short T's. For example, what you just saw can sometimes look like that. You see that one side of the T is really long, the other side is really short. So sometimes they look like that. Because if this was the spinal cord over here, they don't need to go that far to send the signal. But if this other side is the long side, it can travel all the way down, let's say, to the tip of the finger. And we're taking information from there and sending it all the way to the edge of the spinal cord. That's what you see there. The motor, right? Again, impulses from the CNS to the effectors. Those are going to be multipolar neurons. Okay. Most cell bodies are going to be in the CNS for motor neurons that are interneurons, or those association neurons. They're the processing ones, right? Those are the ones between the motor and the sensory. Those are the ones that are mostly CNS, right? A vast majority of our neurons are interneurons, association neurons, the ones that you find in the brain and spinal cord. 99% of them are that. And here's an example of each one. Here's your multipolar, here's your bipolar, here's your unipolar, right? So we don't see the dendrites there, but the red one here, it says motor neuron. We know it's a motor, so it's a multipolar. There's that long axon. The little dot represents the cell body. And you would see the dendrites there are just really small, right? Here's your bipolar. Here's your retina of the eye. That signal being sent from the edge of the eye back in the back portion of it. This would be your rods and cones, right? If you heard this before, where you perceive color versus black and white or gray shades. These are all types of bipolar neurons connecting to then the brain. And then here is your unipolar. Here's your T shape. There's that little, there's your cell body right there forming the T. Can you see that this is longer than that side? Right? Part of the sensory neurons. Okay.